Hi everybody, welcome to week 8. I can't believe we got through to week 8 already. This is my favourite week where we actually get into the applications of what we've been learning about. And one of those is biological chemistry. And we're going to focus on proteins and amino acids because that underpins everything. If you've studied any biology, you'll know about genetics. And the genetics determines what the proteins look like. So the proteins are at the very beginning what determines everything about you. So I'm going to go through this. Hopefully we all learn something. So a little bit of biology. Proteins are enzymes, which we know that catalysts speed up chemical reactions. Enzymes do the same thing, just in biological systems. So enzymes are biocatalysts, or they reduce the activation energy required. So the normal pathway might look like this in activation energy. Enzyme creates a shortcut. So they can be structural components, proteins within the muscles, actin, myosin, cell membranes that make up the structural component that keeps the cells together. So hemoglobin is the important one and the one I'll focus on looking through here. It's made up of 4 different proteins that are all bound together in different ways to make a certain shape. But we can also have antibodies in the immune system. I'm going to keep it simple because there's about 50 slides to this one. So polymers, we have monomers. Hopefully you're reading along with the slides because I'm going to stop sharing so you can see the whole whiteboard. So monomers, 1 subunit. Polymers, made up of monomers. Many monomers. So if we think of other organic compounds that make this, polystyrene, polyethylene, so on and so forth, are all polymers. Now, big hint for your Year 12 exam. Polymers. And if you have me as one of your tutors, then we will focus a lot on these polymers. And the monomers we're going to talk about today are the amino acids. So, monomer, the amino acids, you can guess that proteins are going to be made up of many amino acids. So there's 20 of them. They're organic molecules, so they have a functional group. So they have an amino group on this side. Carboxylic acid group on the other side. And then they have this R group. And this is like with the organic compounds, this is what makes them different. And you have this carbon in the middle, which is the central or the alpha carbon. I'm going to move through these relatively quickly just because there's a lot to go. But that's the joy of the recording. You can stop it and pause it whenever you need to. So, general structure of amino acids. Then we have the R group. So on this side, we have the amino group. Or the amino, yeah, the amino end, I guess, would be a more better term. This, we have the carboxyl. And these are the same on all the amino acids, but it's this that differs. It is the functional group. And if we count the carbons backwards from here, we call these 2 amino acids. So, and this, like in our organic compounds, tells us that the functional R group comes off the second carbon. So, you can look at the next slide and you can see how it kind of looks in 3D space. So if you look at the examples, alanine has a CH3 functional group. And this becomes really evident when we look at proteins and what happens. I mean, I'm not going to try and draw those structures because I'll be here all day, but all of the different amino acids have a differing R group. And the amino acids, so asparagine is ASP, glycine, GLY, so on and so forth. There's 20 of them. If you're looking for all of the amino acids, I'll share the slide and have a look at all of them. Yeah. So over here, well, you guys can just see it on the screen. I can see it over this one. You've got essential and non-essential. So essential means we cannot synthesize or recycle these. We have to get them from the outside world. So that's from our diet normally. We then have non-essential, which we can synthesize or recycle. Sulfur containing, which is cysteine and methionine. I mean, I won't go through every single one and explain because really we're not worried about that. We're looking at the interactions between. Plants manufacture amino acids from nitrogen, water, and sunlight, and nitrates, so the nitrogen in the soil. And then the animals eat those. So if you're thinking about back to your basic ecology, you've got the primary producers on the bottom. The herbivores eat that, convert the amino acids into proteins in their muscles. They're then eaten. The amino acids from their muscles go into the next bigger predator, and so on and so forth. So they say if you're trying to get big and muscly, you should eat lots of proteins because the body uses amino acids to rebuild broken down muscle tissue. So let's get into the chemistry. Good stuff, the boring biology. I love biology, but it can be boring at times. So, condensation reaction. Where's my green pen? What happens when my child comes into the room? Look, guess we're going. Oh, there it is. So, condensation reactions equal production of water. So we have OH here, then we have hydrogen here. Now where might we find these? Oh, conveniently, this is what the amino acids look like on the outsides. So this is how two monomers become a polymer. So on one side we have the carboxylic group. On the other side we have the amino group. So this one we get rid of this, they come together, we get rid of, and a new carbon to nitrogen bond forms between the amino acids. So the OH group on the end of the carboxylic acid, or the C terminus, C side of the amino acid, reacts with the hydrogen on the other side of the next amino acid. Water is released, and then we get a carbon to nitrogen bond. That's a condensation reaction, specifically with regards to the amino acids. So the next slide is just saying we can combine the OH on one side and the H on the other side to produce water. We get a carbon to nitrogen bond. Everyone's happy. So peptides. We're not quite out of protein yet. Peptide is one or more amino acids. Sorry. Two or more. One amino acid is just an amino acid. So if it's made from two amino acids, we call it a dipeptide. And then anything more than that is a polypeptide. So this isn't a protein yet. It's just a string of amino acids held together by the formation of these condensation reactions. And this forms a peptide bond. So peptide bond forms from a condensation reaction between the carboxyl group of one amino acid and the amino group of the other amino acid. Water is released, we get a carbon to nitrogen peptide bond. So I'm not going to attempt to draw that on the board. I'll share it. But you can see you've got alanine, glutamate, glycine, valine, and lysine. So these are the different R groups. Here, some of them are simple, like this guy. Some of them are more complex, multi-chain. But they're all held together by the carbon to nitrogen bonds formed by the condensation reaction, which releases water. which forms the peptide bond which holds the amino acids together. So again, I'm still sharing so you can see OH group on one of the amino acids reacts with the H on the amino group of the other one. We get rid of this because water is formed and a carbon and nitrogen in a peptide bond. And then we can keep condensing and creating a the chain I don't think can be never ending, but effectively it can. So I think I got through that quicker than I thought I was going to. Whoops. So proteins. So amino acids. Singular. So amino acid is just one amino acid. A dipeptide. Two amino acids. Stop sharing, so. Polypeptide. Polypeptide is 3 to 49 amino acids. And a protein. +50. So we know that proteins are biopolymers because they're made up of multiple subunits or monomers. Proteins are made up of amino acids. So here's where it gets interesting. Back in the day when they wanted to diagnose diabetes, the doctor used to smell and taste the urine sample to see if it had high levels of glucose. And the reason that diabetics have high levels of glucose is because of the hormone insulin. It's relatively simple. It's got 51 amino acids, so it's not very big. And before the advent of the molecular biology revolution, we used to take the pancreas of pigs and other animals that were sent to the slaughter and we would extract the insulin from it. 48 of the 51 amino acids are the same in this sort of porcine. So all three of them are virtually the same, aside from a couple of amino acids. So what we used to do, we would take that insulin, give it to the diabetics, and they would inject it. And it works okay for a little while. But the body then, after a couple of years, will reject that, and then the nurse can no longer be treated for diabetes. Now, just remember this as we go through. It is almost identical to the human insulin. But the body rejects it because it knows that it's not the same. And it doesn't work as well in the human body as human insulin would. They are almost the same at this level. So remember that as we go through. So let's look at the structure of the protein. There's 4 layers or structures. So we have the primary structure, So we want to be able to explain primary, secondary, tertiary, and quaternary. Yeah. I've been teaching this for years, and I always mispronounce quaternary structure of the protein. So the first layer is the primary structure. Hopefully you've got the slides going along. And the only difference at this level of our insulin, two different varieties, human and porcine, is the sequence and type of amino acids. So this is what we call the primary structure. And I've skipped ahead a slide because I'll wait till the end to show you that. So the primary structure is the very base level of the structure of the protein. So it's the number of amino acids, the type, and the sequence of the subunits or the monomers. And we start the primary structure on the N terminus, so where you have NH3, and then we add to the right-hand side of this. In these condensation reactions is how it gets built. And then at the end, we have the C or the carboxyl end. And all the other amino acids slot in here. So our insulins, only difference is 3 of the amino acids, plus the sequence is not the same order, but it's still there. So this is the free amino group. The free amino group. And this is the free carboxyl group. This guy's just hanging out here on the end by himself. He's attached here, sorry, he or she. So they've got these carbon to nitrogen bonds all the way through here, where the amino acids are. So it starts, again, a good way to remember it. It starts at the amino group, or A. A comes first in the alphabet. It finishes with the carboxyl group. C is after A in the alphabet. So this is just going over everything. Proteins are long polymers made from 50 or more amino acids. They start with the N terminus and the C. Amino group, we know it's an N terminus because it has nitrogen. C terminus has carbon, another cheap. And we have these carbon to nitrogen peptide bonds caused by a condensation reaction. So, primary structure is the simplest view, and it doesn't really tell us much. We can't go from this structure, put it into a computer, and suddenly we know exactly what the protein looks like. We don't have enough computing power on the planet at any one time to be able to do that. It would take us decades. And it probably still wouldn't be right. So let's look at, I'm not going to attempt to draw it on the board because we all know how that goes. So we've got the N terminus here, we have carbon to nitrogen bonds all the way through. On here, we have the C terminus. Because that's the end, you've got a free carbon to oxygen double bond. You can see the differing R groups here. So cysteine, arginine, valine, asparagine, I can never remember early, and cysteine here. So you can see they're all exactly the same except for these R groups. And that becomes really important when we're looking at protein folding. So this is the protein structure in a straight line. So and this, although we can't actually figure it out just based on this in the entire 3D structure is dependent on the sequence of these. And if the body doesn't get this right, so there's lots of genetic diseases where one of these amino acids has been changed or deleted. So a change in one amino acid can tell the body to stop the production of an embryo. So if it senses there's one amino acid change in this protein, that embryo will no longer be seen as viable. So it's very, very important that the body gets it right. And we can use protein sequencing to determine the primary structure, but it doesn't tell us a huge amount. So there's bovine insulin, which again is almost identical to human insulin. But if we use it, eventually the body will reject it, and it doesn't actually work anywhere near as well. And that's because of the structure, and I'll go through that as well. So this is where it starts to get interesting. So we have a straight line. So we have our primary structure, which is in a straight line. But in this, there are positively charged hydrogen. And over here, there's on another group, there might be negatively charged oxygen. So this protein will do this and form a hydrogen bond between the NH and the carboxylic acid group of another one. So it begins to fold. And this is what we refer to when we're talking about proteins folding into shape or folding into the wrong shape and causing diseases. So proteins, or these amino acids, always have these groups. And we know that nitrogen and hydrogen is polar, so it's always going to have positive hydrogen, which is left floating out here. Carboxylic acid is always going to be a negative dipole with oxygen being minus. So we get a hydrogen bond being formed, which is really strong. We also, in this protein, we have all kinds of things, dispersion forces, dispersion forces, dipole-dipole forces, hydrogen bonding, ionic bonding, covalent bonding, and all of this works together to determine the 3D shape. So if one of these straight line, if one thing is missing or is wrong, then this won't fold into the right shape. which if it's an important enzyme that's supposed to be a certain shape, then the protein, well, that person will be unable to regulate. So let's say cystic fibrosis is a genetic disorder, affects the protein that tells the body how thick to make the mucus. That protein doesn't fold properly, and unfortunately those people suffer lifelong. But it's only a change in a couple of amino acids that completely throws off the shape. So we have, when this starts to fold, we have alpha helices and B or beta pleated sheets. So the alpha helix looks like this. Obviously, the drawing on there is much better, and it's formed by these hydrogen to oxygen hydrogen bonds that form, and you can see there on the slide that the chain then is held in shape by those strong hydrogen bonds. We also have beta pleated sheets, which are these weird, like, I don't know, look like foil that's been folded into that shape. And that is when you have two or more polypeptide chains, so two more of these, then this guy comes along and they bond together. That's only when there is more than one subunit within the protein, so more than one primary chain. So you can see there the polypeptide chains folded over multiple times, and that holds the structure together. And it looks weird, and it's really hard to conceptualize. So the tertiary structure is really important. So tertiary structure is the overall 3D shape of the protein. So you can see the secondary structures, and that's what that protein will look like in a 3D space. You can see the NH2 group on the left, or the amino group. And then you've got the COOH on the other side. You've got beta pleated sheets and alpha helices. So that one's mostly alpha helices because it's just one polypeptide chain. So there is another, I won't say the word thing, another, what's the word, factor to this. So it's the interactions between the R groups on the amino acids. Some of them are negatively charged, some of them are positively charged, some are hydrophilic, some are hydrophobic. Some contain cysteine, some are ring-shaped, some are aromatic, so on and so forth. But the different amino acids can interact with each other and form, remember, all of our intermolecular bonds. And once they're in that secondary structure, because that's to do not with the R groups, but just the bits of the amino acids that are common to all of them. Now we're looking at the R groups. So the R groups can further twist the protein into different shapes. Through hydrogen bonds, dipole, and dispersion, as well as ionic interactions. So ionic being ions, so electrical charges. So we have on one of the amino acids, we have COO minus at the end of its R group. The other we have NH3. Sorry, NH3 plus. And because there's a negative and a positive charge, they interact and the protein will twist in that 3D shape to further fold down. And the whole point of the protein folding down is to reach the most stable state. So atoms will interact with each other to reach the noble gas configuration. Proteins will fold so that there's no possible way that it could unfold. So it will react all of the negatively charged ends and positively charged ends. It'll react everything together until it's stable, until it can sit there and nothing happens. Or the lowest energy state. We also have disulfide bridges. And that's when we have cysteine has sulfur in its R group. Sulfur in R group. And what happens is you get a covalent bond between sulfur and sulfur, again, making the protein twist and change in that 3D shape. Overall, what we call the conformational shape. So we're now getting to the tertiary structure. So this disulfide, if you've got no sulfur here, it will actually turn into something like that. So we will completely change the shape there. So you can see this disulfide bridges. Whenever you see cysteine and cysteine or CYS, you will get these disulfide bridges. And I think one of the big differences between bovine and human insulin contains 4 cysteine, if I recall correctly. So, tertiary structure. And this encompasses everything before this. And this is the final shape of the protein. And this is why the amino acids being the same is so important, and why the change in one amino acid can have catastrophic consequences. So we have the tertiary structure. So this takes into account the secondary structures, the alpha helices and the beta sheets, the additional bonding that takes place, so the extra hydrogen bonding and the extra covalent bonding, and then the interaction between the R groups. So this is when, if this structure doesn't work or isn't the same, if it's an enzyme, I'm going to stick with enzyme because I might do an extra talk on enzyme, because that was the third part of this series. Enzymes, it's supposed to be this shape. You change one amino acid and it becomes this shape, then the enzyme will not work. So most of our genetic disorders are caused by a change in the primary structure, which alters the secondary structure. And because we've changed amino acids, this alters the tertiary structure, what it looks like in 3D space, like that. And in here might be the binding site for the enzyme. In this bit here, if it's not the same, and this changes shape ever so slightly, enzyme is completely useless. And we can confirm that by when we try to use porcine or bovine insulin in humans. It's very, very, very similar, but it just does not work the same. And then we have the quaternary structure. And this is only for proteins that have more than one polypeptide. Good way to remember. Most of them have 4. So we have 4 of these. And you can see that the number of interactions that could be possible gets infinitely complex the more you add. The most common one is hemoglobin. It has 4 subunits. So you can see the alpha helices and the beta pleated sheets, and everything's twisted into place. For all 4 of the tertiary structures, 2, 3, 4. And then the quaternary structure looks at how all 4 of those chains will fit together in 3D space. Again, if you slightly alter any of the amino acids in these subunits, then the quaternary structure will not be the same. And the shape will be off. If there's more than 4, if there's more than 1, we go to the quaternary. If there's only 1 subunit or peptide chain, then it doesn't go past the tertiary stage. It's only 1. Anything above 1 goes to quaternary. So, quick recap. Primary structure is the sequence of amino acids in the polypeptide chain. Secondary is the change in shape because of the hydrogen bonds between the amino groups and the carboxylic groups. Tertiary is the overall 3D shape of the molecule. It's mostly influenced by the R chains and the bonding that happens between them. And quaternary is if there is more than 1 protein chain that's involved. So I've sent this out. These are the questions for this week. So yeah, I've sent them all out to you. You should have been watching along with me, hopefully. And I'll see everyone next week. Thank you.